Hey there, welcome back to the Atlasapprox. Today I'm going to show you how to use our interface to identify the highest expressors of your genes of interest across different organs and cell types in any species. Maybe you feel a bit confused why we need to focus on the highest measurements. Well, first, it allows us to identify the key cell types that are most actively expressing specific genes providing insight into cellular functions. Second, it enables us to compare gene expression across different organs. Then we can see the variations in gene expression throughout the body. Finally, examining high expression levels in certain cell types can offer information about the potential roles and functions of these genes, contributing to a deeper understanding of their biological significance. Before we dive into examples, let's talk about some query types you can use to explore the data. The first query type focuses on the single gene analysis. This lets you determine where a specific gene shows the highest expression. The second type can be used on the gene list analysis. With this, you can analyze the combined highest expressors for a list of genes. And then our advanced query allows you to explore which cell types express one certain gene higher and then other specific genes relatively lower. These query types give you flexibility in how you approach your gene expression analysis. In this tutorial, we are going to look at three examples, each showcasing one of these query types. Let's start with our first example, using the single gene analysis query type. On the landing page, enter the following query in the search bar. What cell type is the highest expressor of CD19 in human? Hit enter. And then our interface will generate results, including a list of top expressors and an interactive body map below on the left. The bar chart on the top shows gene expression levels in different cell types and organs. The organ map is color-coded to show where CD19 expresses actively, with darker color indicating higher expression. For additional features, we can hover over an organ on the map to see a detailed breakdown of expression in different cell types within that organ, as the chart presents on the right. Now, let's move to our second query type, gene list analysis. Enter this query. What cell type is the highest expressor of FOXP3, REG1, REG2, CD28, CD68 in mouse? The results will include a bar chart showing the combined expression score of these genes, and then a dot plot with genes as rows and then cell types as columns. In the dot plot, bigger and darker dots mean higher expression and more cells expressing the gene. Also, we can hover over dots for more details about gene expression in specific cell types. Now let's look at our advanced query type. Enter the query. Who express COL1A1 but is missing Gzma and Gzmb in mouse? From the output of the interface, this bar chart shows the expression score of cell types in mouse that highly express COL1A1 while with low expression of Gzma or Gzmb. Each bar represents a cell type in an organ, and then the higher the score, the stronger the expression of COL1A1, and then the lower the expression of Gzma, Gzmb in that cell type. In the dot plot below, we can see the specific cell types and organs where COL1A1 is highly expressed and then GZMA, GZMB are expressed relatively lower. For example, fibroblasts in various organs, such as in muscle, fat, kidney, strongly express COL1A1 more than GZMA or GZMB. Other cell types, like pericytes in fat, also show this pattern. And there you have it. You've just learned how to use the query types of the Haggis measurement feature in Atlasapprox. Feel free to experiment with different genes, species, and query type. I'll see you in the next tutorial.